Welcome to HSMD Research, or Human Somatic Mutation Database for Research, designed for academic researchers working on variants related to top cancer genes. Keep watching to see what awaits you. Even though this free access is limited, the genes and information that you can access are applicable to a number of cancer types, such as breast, skin, colon, lung, thyroid, etc. This is the homepage for HSMD Research, which shows the amount of information available to users. There is limited access to drug and trial-related information, which I'll explain as we go along. From the homepage, you can click on Explore Genes, which will take you to the Gene Explorer page. Here, the names of the 25 genes that are included in HSMD Research are hyperlinked, written in blue and underlined, such as PTN and ALK. Genes that are not included are grayed out, such as POL. And when you hover over their names, you will see a tooltip saying that more information is available through a subscription. But don't dismiss this page just yet. This alone already shows you a wealth of information, whether the gene is included or not, such as the distribution of the different alteration types and functional impact, whether the mutations are known activating or predicted activating, etc. You can also easily see the number of diseases associated with the gene and the actionable distribution of the variants seen across the gene, such as how many are Tier 1 or those with FDA-approved drugs, and how many are Tier 2, which have recommendations from clinical practice guidelines, as well as Tiers 3 and 4. Clinical significance is also clearly indicated. If any variant in the gene is associated with sensitivity, there's an S, P for prognosis, D for diagnosis, and even therapeutic resistance, which is denoted by R. You can also get a general idea of the number of drugs associated with each gene on the list, and the total number of variants, or alterations. Now let's dive in by going into the entry for ALK. Right off the bat, you can see different sections with anchor links at the top of the page for easier navigation. First, you have the description, which tells me that ALK stands for anaplastic lymphoma kinase, as well as its main functions, other information, and the references these are sourced from. This is followed by a section on external links, which includes QIAGEN links and third-party databases. Using the anchor links, let's jump to the alteration type and functional impact table, which has hyperlink numbers. Clicking on one of these will redirect you to the alterations table and apply the option that you chose as a filter. Moving back, you also have actionability distribution, which also works in tandem with the alterations table, followed by therapeutic significance, which is available for paid access. Now, let's go to clinical trials. This shows our extensive list of curated clinical trials with the relevant biomarkers specified, together with all these clinical trial information, drugs, the disease specified in the trials, phases, and more. References require paid access, which also applies to the next sections of diagnostic and prognostic significance. However, to give you a clearer picture, these sections feature a table of diagnostic or prognostic tiers, and whether they are mentioned in any of the clinical practice guidelines. Then we have the observed clinical case distribution section, showing which mutations of ALK are most prevalent in different cancer types, which, if you think about it, reflects the current clinical landscape. This could come in really handy for academic labs. For example, if you're looking for the best preclinical model to use for your research, you can use this section to find the most appropriate and best fit cancer type to do your experiments on. Up next is alteration distribution. In this graphic, you can see along the protein's span the regions where you can observe the most frequent mutations and hotspots. Let's scroll down to the alterations table itself, which shows an extensive list of all the alterations that have been observed in this gene, along with the alteration type and functional impact. Let's clear the filter for now to see all of the results. As you may know, it typically takes a long time to gather this information, but HSMD Research gives you these details right away. You can see if a variant is activating, inactivating, or predicted activating, or inactivating. You can also see the actionability information, clinical significance, and the source, which could either be clinical reviewed or QKB literature. Clinical reviewed indicates that this variant has been seen in one or more of our clinical tests and has been reviewed by a variant expert, while QKB literature indicates that the variant was curated from scientific literature. also by a variant expert, yet has not been seen in any of our clinical tests. Next, you have the isoform section, which lists all the isoforms associated with this gene, including the main status column, which helps you choose universally accepted transcripts. Further down, the drugs table shows a comprehensive list of drugs that have this gene, ALK, indicated in the case as a target. If there is evidence in the literature for responsiveness, it is shown together with the sources. In the sources column, clinical studies means that the information has been curated from clinical studies, while FDA means that the drug is FDA approved. If it is curated from clinical practice guidelines such as NCCN or ESMO, it will also be indicated. You can also see the diseases associated with each of these drugs, either in the context of approvals or clinical studies. Finally, you have all of the information that went into the curation of this data in the bibliography section. Note, though, that for HSMD Research users, only the top 15 references will be shown. In addition, while each entry is expandable, only paid users will have access to the listed curated findings from these publications. Aside from the Gene Explorer pages, you can go into Alteration Explorer if you want to focus on a particular variant. This table shows information similar to the Gene Explorer, but this time at the variant level. Again, you have functional impact and actionability tiers based on AMP, ASCO, or CAP guidelines, which are the standard in the field of somatic oncology. You also have clinical significance, as in the Gene Explorer pages. If you want more information on a specific variant, you can click on the hyperlinked coding change or protein change value. When the variant entry opens, you can also see sections with anchor links, similar to the Gene Explorer pages. You also get the description and external links, followed by the alteration details, which lists the chromosome and position numbers and all of the other information you see here, along with an extensive list of transcripts that may be available for the gene and variant. Similar to the Gene Explorer page, information on therapeutic, diagnostic, and prognostic significance are limited to paid users. However, in the clinical trials section, HSMD Research users can access a wide variety of clinical trials that are curated for this specific variant. Next, you have the observed clinical case distribution of the different cancer types involving the specific mutation, including those we've seen in our clinical tests. Sections unique to these Alteration Explorer pages are allele frequency, which includes a link out to Nomad, and biochemical impact, which is based on various predictions coming from these different resources. The drugs and bibliography sections mirror their Gene Explorer counterparts. Last, but certainly not least, let's go to the Disease Explorer page. The entries on this page are completely accessible to HSMD Research users. Our exhaustive list of almost 1,400 diseases has since been organized into a consolidated hierarchy. As you can see, you can expand parent disease nodes to view the child nodes for each of these cancer types. OncoTree codes and SNOMED links are also available. You can also easily see the top 3 mutated genes in any of these cancer types and subtypes. As you go about exploring HSMD Research, remember the rule of thumb. If it's hyperlinked in blue and underlined, you can click on it. What you don't have access to, our tooltips will remind you. That about ends our HSMD Research walkthrough. Thanks for watching.